Okay, so I guess welcome to our first Zoom class. Before we get into things, just want to talk a little bit about Zoom and a little bit about Zoom etiquette. So you may or may not be familiar with using online learning systems, these teleconferencing softwares and all that, but a couple of things to keep in mind. Being on time is always good, whether you are virtual or in person. It's always just a way to be respectful. Always be respectful no matter what you're doing, whether you're asking questions or using the chat, always a good thing as well. So I have Zoom configured so that your microphone and your video are muted. You can ask questions at any time using your microphone. You'll have to unmute it. But I do ask that people, dress appropriately. You never know if you press the wrong button and you actually turn your video on. I don't want people to be, I don't know, let's say in their bathrobes or worse, or somebody doing something weird in the background or whatever. Dress decently, and there have been people who have made that mistake and had some embarrassing moments. So we don't want that to happen. So I think I mentioned the microphone already. Just keep it muted unless you want to speak. Otherwise, we'll hear everyone's cough and sniffle and dog and cat and whatever else is going on in your place. You can ask questions using the chat function. So there is a chat function in Zoom. If you go to the Zoom bar, you'll see a chat function. I see at least one person said hello. And so I'll kind of have the chat function running at most times. And if you have any questions, you can always ask questions there, and I'll take a look at that from time to time to make sure I can answer any questions. And come prepared to learn, just like any class. Come to learn, and we'll do well. So for the most part, what I'm going to do is I'm not going to have my video running, so I'm going to turn that off now just to save some bandwidth, and that way we can focus on the class material. So I apologize for this. I can't control the pandemic and what everyone else decides. I know that the in-person learning experience is quite a bit better, but I'll do my best to make the Zoom classes interactive and as interesting as they would be if they're in person. So still allowing some people in on time. Please try to join early. It helps everyone else, so I'm not clicking all the time and trying to allow people in. Okay, so before we get to today's topic, I wanted to talk a little bit about the labs and talk a little bit about what's expected of you for Thursday, because you're going to have your first report due on Thursday. So the first tip that I want to give everyone is how to answer questions. So you can take a look. Here's a question here. I think we have the same one this year, and you can see it's worth 3 marks. So what do I mean by 3 marks? Maybe 3 sentences, maybe 6 sentences, probably not 1 sentence. 3 marks is kind of 3 points, but not like 3 words, right? So I want to have you be thorough, use good scientific language, be specific, be quantitative, all those kind of things that make good scientific observations and so on. So I have a couple of examples here, or at least a start of an example. I may have the same example in the lab video I sent you on Thursday. But here's a couple of sample questions, or sample answers. So you can see it says here, which is better? The doorknob had the most bacteria and the floor the least. Or the second one, the swab of the doorknob of the chemistry lab produced 55 colonies, representing the tested area with the greatest number of bacteria. So you can see the second one, I would say is better, and it doesn't mention the floor, but that would probably be another sentence. And you can see some of the things that are helping it. So you can see we're talking about, it's a little more specific about exactly what was swabbed. What does this mean, most and least? I mean, you could be talking about comparing 2 and 3 or 205, versus 5. 55 colonies tells me exactly how much is there. It's very, very specific. And that's what I like to see. I want to be able to read your responses and not have any questions in my head. I want it to be very specific. You can see the other parts of the question is actually talking about diversity. So diversity means, what are the differences? You could talk about things like color, size. I noticed some people had some mold in their plates, so you could mention that. It's kind of fluffy looking and all that. So if you have any questions about that, let me know. But I do find myself writing that comment a lot on people's reports about that they're being kind of vague and not very specific. So you've been warned. So people want to know what they need to do for lab 1-2. I'm hoping this has been very clear, but I'm just reiterating it again. You need to basically, for the first short report, is you're answering these questions. So these are on pages 37 and 38 of the lab manual. And we're not going to do question 2 because lab 2 was canceled, but all the other questions you can answer either using information from lab 1 or there's the lab 3 pre-lab. I'll talk about that in a moment. So you can see there's a question here about using a microscope. There's a question here about reproducing a scientific figure, the one that you drew in lab 1. And then question 4 is asking you about those bacterial plates. You can see it's worth 4 marks this time. I like to have things kind of add up nice and neat. It's supposed to add up to 25. So I'll have to adjust that somehow for the final grade since we're skipping that one question. So let's talk about this lab 3 pre-lab assignment for a minute. So this is the only pre-lab assignment that you have. It's for lab 3. So lab 3 is the big lab, the one that we have to do a formal report for. And so this is to help you get ready for a formal report. I'm not going to go over all the details for this. I sent out a video last week in the email. I sent it out on Thursday. You can always go find it on the Moodle site as well. And so this lab 3 pre-lab assignment, you can see there's a few questions pertaining to that. So what you're going to do is learn how to use a website called PubMed. That's a journal database. And you're going to find a scientific paper and you're going to learn how to reference and cite it. So like I said, go watch the video and it will give you lots of information on how to do this. And you're going to need to do that for lab 3. So one of the things you'll be submitting for lab 3 is a journal article for this pre-lab that you're going to find for me. So how to hand in your report. As I mentioned before, I'm doing all digital submissions this year. So you're going to have to go to Moodle, scroll down to Laboratory, and click on that little hand in link. And you go there, and there's a place for you to upload your file. I'd like to type up your assignment as a Word document. And it's not necessary, but it certainly makes me a lot easier for me to find everyone's reports if you put your name in the file name. So if you could, I would prefer you to use a format that looks something like this. So tell me the course, tell me what the assignment is, and tell me your name all in the file name. All those things make it much, much easier for me. I know sometimes I end up with these things that are untitled or it's just lab 1. Well, I do teach other courses and I do keep things pretty straight, but anything that helps me is going to help you, right? Because that means I'm in a better mood when I grade your report. So you are going to have your diagram that you're going to do. You can scan or photograph it. Make sure it's a high quality scan or photograph. It's blurry or something, you're going to lose marks for that. You can embed it directly into the Word document. If you don't know how to do that, you can ask me. I can show you how. It's very easy to do. You can submit it as a separate document. And your journal article will be a separate document as well. So I'm expecting 2 or 3 documents for lab 1 and 2. So that's it about the lab for now. And just want to remind everybody that for the topic on viruses, I made a recorded lecture and I sent that out on Friday, and just looking at the number of views, not everyone has actually looked at it yet. So don't want people to get behind. So do make sure you do look at that. Take some notes. I have a quiz on Moodle for that already. All right, so today we are going to talk about mostly microscopy and probably mostly light microscopy. So let's get into that, because this is a really important tool that's used for cell biology and many types of modern biology. And kind of unfortunately the way that these lockdowns have worked, we're missing a lot of our microscope labs, which is really unfortunate. But at least you had some experience for lab 1. So one thing that I talked about in the first lecture was that things are small. You can see this is showing some things that we can see with the human eye, but it does not include a lot of cellular and subcellular structures. Maybe frog eggs and human eggs are kind of, you know, they're going to look just like very, very small specks. So most of what we're looking at for looking at cells we're going to be using light microscopes or sometimes electron microscopes. And there's one other type of microscopy I'm going to talk about in this class, which is called scanning probe microscopy. So we'll compare and contrast those things here as we get into it. So let's talk about a microscope. We're talking about basically 3 basic things here. So for a microscope, you're going to have a specimen. Hopefully that one's obvious. You need something to look at. For 2, you need some sort of, we're not going to call it a light source because electron microscopes don't use light, but we're going to call it illumination source. We'll call it illumination source. And 3, you can see on there some sort of lens system. So there's my little object there. And guess what I didn't draw on this diagram is an illumination source, but we can maybe picture there would be, I'm going to draw a light bulb here. There's my light bulb. And the light, of course, is going to go through or bounce off the specimen or something like that. My slide doesn't seem to be lining up for some reason. It's going to go through the lens. The lens is going to take the light and manipulate it in such a way that it's going to produce a much larger image. So there's an example there, and it's a very, very simple kind of microscope. So we did talk about one of these simple microscopes. So this one here was the one we talked about in the first week of classes. This was Antony Van Leeuwenhoek. I'm not going to attempt to spell his name, but it's V-A-N-L and a bunch of other letters. You never remember where all the vowels fall in. And as we mentioned before, this is basically a lens, and the illumination source was the sunlight, and everything else is just an elaborate contraption for holding on to the lens and focusing the specimen. It didn't take long before people started to make compound microscopes. So we'll talk about them in a minute. But the whole idea is that you have two lenses. You have one lens down there and another lens up here. And so this is what Robert Hooke apparently used, something like that. This little apparatus here is an oil lamp. And so that's for the illumination of the specimen. And most modern microscopes have multiple objective lenses. So compound, by the way, it means more than one lens, right? So this one here up on the right, it has an ocular lens over here, and it has 3 objective lenses down here. So you can zoom in or zoom out based on the size of the specimen or whatever details you're trying to look at. So here's kind of an inner diagram of your typical compound light microscope. You probably realize that it's a bit more complex than that little black one there. And so what you're looking at here is you've got a series of things to either make a better image of the specimen, or either that or just make it a little more ergonomic. So if you take a look at this one here, you've got this little prism here, for example, and the prism is basically bending the light, and it's coming out at a different angle. Otherwise, you know, your eyeballs are going to be stuck in there at a really weird angle, and it's going to be difficult to take a look at that. Other things, oops. I see there's a couple questions. I'll come back to that in a moment. Other things is you've got a nice bright luminar light. You've got these condenser lenses that are used to focus the light. All those things are going to help with the image a little bit. All right, sorry I did miss those chat questions. Couple of questions here. Somebody says, should the calculations of the diagram be that of the one on the computer or the actual document that you make? Because sometimes I guess the whole idea is you're asking if you scan your photo and then it gets resized in Word. I'm fine if you do it with the original diagram. If you scan a whole page, it won't make any difference at all, but I'm fine if you have your calculations for the original diagram. Someone's asking about the video for the virus lecture. I emailed it out on Friday. You can also go to Moodle, and at Moodle I've linked the playlist for this class, and you can find the virus lecture there. So I just want to talk a little bit about, I kind of meant to show this before our first lab, but I'll play this for you now. And this is just talking about using a light microscope, and talks a little bit about the different objective lenses. So I'll play that for you right now. It's a pretty short video.
In this video,
sorry, I just had to make sure I had sound going. All right, let's go.
I'm going to demonstrate how to focus the microscope. Begin by turning your microscope on and adjusting the light intensity to about 5 or 6. Then place your slide in the clamp on the stage and center the specimen over the condenser. Then turn the low power objective into place and use the coarse focus to move the stage all the way to the top. While looking through the objectives, slowly turn the coarse focus down until you can find your specimen. When you think you've found it, wiggle the stage a little bit to make sure what you're focusing on actually moves with the slide. If you wiggle the stage and nothing moves, you're focused on the wrong plane. Once you've focused at 10x, you're going to rotate the next power into place, the 40x. This time, use only the fine focus to adjust the stage and focus on your specimen.
So I'm gonna stop there. You can always come back and take a look at this later. Like I said, I think it would have been more useful for me to play this before the first lab, where we're actually using the microscopes. But you can see she's showing the different objective lenses, and I think everybody already had some practice using those. So I want to kind of talk about, how we use microscopes and what kind of things that we can see. There's the one that, of course, we were using in our class. So one thing that just pains me, the labs that we're missing here. You guys are not going to get to do the bacterial lab in person next week. And in that lab, we use something called oil immersion. And there's kind of a problem with microscopes as you zoom in. So if you take a look at this, I want you to just take a look at this, this red light here, right? So light, as it goes through different mediums, sometimes it bends. And you probably notice this if you, look through water and things kind of, the light looks warped and all that. And so you can see this red light here, it kind of, as it goes from glass to air, some of the light scatters in all sorts of different directions. And so the problem is when you zoom in on something using a really big lens like that oil immersion lens, you don't get enough light. So what you can do is you can actually put a blob of oil, you can see it's called immersion oil, right on top of the microscope slide. And so instead, the light will pass basically more directly through the specimen, and you get a lot more light. And this is something we would have used to look at bacteria. And I'll make a little video on this for lab 4, because unfortunately lab 4 will be virtual. So let's talk about what we can do for microscopes to get good images, because ultimately this is what we want. And I think I'd mentioned in the lab that our microscopes are maybe $8,000 or $10,000 or something like that each. So why is this one so good compared to, let's say, I don't know, you can probably get a plastic microscope at Walmart for 50 or 60 bucks, right? So why didn't we get all the Walmart microscopes and save a whole bundle of money? So one thing that is good is to have magnification. And so magnification is basically the size the object looks compared to the actual size of the object, right? So you're let's say magnifying it 1,000 times or 3,000 times or something like that. So how do we improve magnification? You get a bigger lens. So if you take a look, here's our objective lenses down here, and you may have noticed that some of them are bigger than others, and the bigger lenses do have better magnification. It's kind of like the same thing if you have prescription glasses. You probably know that somebody with thicker glasses has a bigger prescription in order to magnify whatever it is, magnify the world a little bit better so that they can see a lot more clearly. So you can still get different magnification in a Walmart microscope. Oh, there's the lenses right there. So you can see their biggest ones are on the left and the smaller ones on the right. So you can still get different magnification, but it's kind of like you probably know if you have an older cell phone and if you zoom in, the picture doesn't look as nice, but you can still zoom in and get a big image. But newer cell phones tend to have better lenses and they have better zoom capabilities. So something else with the image is besides magnification, sometimes we want things to be sharp. And so sharpness or resolution is really how clear it is, or clarity is another good word for resolution. And we're talking a little bit about resolution in lab 1 when I had you fill out that one table. So you can see I have a definition there. It says it's talking about the minimum distance of 2 distinguishable points. So take a look at this. Here are 2 images that are magnified at the exact same amount. But if you take a look at the one on the left, it's kind of hard to tell where one cell finishes and the next cell begins. Whereas the one on the right, you can definitely make out each individual cell. And so the one on the right, you can see it has a resolution of a much smaller number. So 0.2 micrometers versus 2 micrometers. So this one here has the better resolution. Has a smaller number, it means it can see smaller objects. So resolution is clarity. Far side cartoon for you. You can see it's some amoeba. They're looking at a little family slideshow, and he says, "No, wait, that's not Uncle Floyd. Who's that? I think it's just an air bubble." So clarity, resolution. So how do we improve resolution? One of the big things we do is the quality of the lens material. So I'd already mentioned what that Walmart microscope might be made of. Probably plastic. Plastic lenses tend to not be as good as more expensive materials. So glass is a lot better than plastic, and quartz is a lot better than glass. So the microscopes that we have in the lab are quartz lenses. I can't remember how much each lens is. I think it's a few $100s just for one lens, never mind the rest of the microscope. And sometimes we can play around with the light as well, using ultraviolet and things like that. But the big one is the lens materials. So we want to improve our image quality. We can make it bigger. We can improve the resolution by making it sharper and having better lenses. And the last thing that we can do is improve the contrast. So contrast is basically that you can tell the difference between different parts of a sample. So I'll show you a good example of some contrast. And we can do this by staining. So we'd be doing the ground stains in lab 4. So if you take a look at this, we have on the left, we have some cells. We can actually, this is a very good diagram or image. You can see a lot of details there. But when you stain it, it just pops out. And so sometimes without the stain, you can barely see anything at all. So stains tend to be charged molecules. And you probably know from your chemistry that opposite charges attract, right? So for example, DNA is negative. And so a lot of stains that bind DNA would be positively stained molecules and so on. So very, very useful to use staining. So there's another image. This is Giardia. This is a protist that has 2 nuclei and kind of looks like a little bit of an alien. I thought they're kind of cool to look at. So I want to talk just for a minute about the gram stain. We're going to come back and talk about the gram stain in our unit on bacteria, and we'll talk about the gram stain in lab 4. The gram stain was invented by a guy named Graham. So Hans Christian Graham. So whenever you see Graham, there should be a big G because it's a person's name. And he was staining bacteria and found that there were 2 different types of bacteria. In fact, some of them would stain purple using his protocol, and so he called them gram positive. And some would stain not purple, so he added another color, and you get this pinkish color, and so we call this gram negative. So we'll talk about, like I said, the gram stain and bacterial cell walls in our next unit in lectures. So I want to just switch here and talk a little bit about some other types of light microscopy. And So I'd mentioned that sometimes you can get better images by using ultraviolet light, and sometimes we have other types of techniques we can use to really make images pop out. The one way to do this is to use fluorescence. So I'd mentioned we have our staining, right? And so what if you had a stain that actually glowed? So that's what fluorescence is. You hit something with some light, and usually it's another color comes off and it glows. So now we can have glowing samples, and we can get some really stunning, amazing images. We're talking about more expensive microscopes. A fluorescence microscope, a very basic one, is $20,000-$25,000. And some of these confocal microscopes here, you can see it says confocal microscopes use lasers. And these lasers can focus on things really precisely. And now we're talking about, I think, a minimum of $50,000, expensive software and things like that. But you can do some amazing things with these confocal microscopes. I'll show you some images. So there's an image of some bacterial cells, and you can see they just pop out nice and nice sharp there. There's some epithelial cells, and you can see they've used two different types of stains, one to stain the nuclei one color, and the cytoplasm another color. And amazing images, really, really pop out. There's our Giardia again. You can see the little spots are I think they're ribosomes, and the two nuclei are looking like big eyeballs. And there's another one, spindle fibers and chromosomes. So some incredible pictures we can get using fluorescence nowadays. Fluorescence is starting to get used a lot more in the medical industry. Microscopes are a lot more expensive, but you can diagnose very quickly. So here's a stain that's looking for Plasmodium, which is the organism that causes malaria, and pops out very easy to see what's in there. So I saw this one on the internet, thought this one was kind of cool. Somebody, I don't know what they're doing, made some sort of rainbow cell, but very, very nice to look at. So I'd mentioned, I just want to point out, right, like I said, there's fluorescence and there's confocal. Confocal really is a type of fluorescence, but it with the lasers, it can focus a lot more precisely. And so you can see here's just comparing the two, and you can see you get often much sharper images using confocal. But again, much more expensive microscopes and softwares and those kind of things. So I want to show you some other cool pictures. There's just some amazing ones out there. This is a competition put on by Nikon, the camera maker, and they have a little competition every year for best photos. And just thought I'd show you some here. Here are some skin cells, and you can just look at those details. They're really, really incredible. There's another one. This is a parasite. This is a tapeworm head. And it looks like something right out of a science fiction movie. Absolutely terrifying. And here's some axons in some neurons, some ganglion from I think it was a chicken or something like that. And not really sure how they had them come out all these different colors, but very, very stunning, very interesting image. So one thing we can do with fluorescence as well, I mentioned this expensive software. Sometimes people will take dozens of photos, and they can kind of merge them into sort of an average and enhance things. You can see there's some sort of dividing cell, and you can see the enhanced image is a lot nicer too. Another thing confocal can do is get three-dimensional information. So there's a budding yeast cell, and you're looking at a mitochondrial network, and really cool stuff. All right, so a couple more photos here, but what I wanted to do, so this is holiday lights, and here's up close and personal. There's the nucleus, and all those are little mitochondria, those little yellowy things. All right, so before I get any further, what I want to do is usually what I'm doing as I'm talking is I'm making some notes on the board. So what I'm going to do is I'm going to make some notes for you right now. And you can see this is talking about how we can improve our images. So magnification, bigger lenses, resolution, better lens material, contrast, staining, and there's other tricks we can do. But it says here that a lot of these organelles are too small to get a lot of good details using light microscopy. So I want to talk about electron microscopy, but before we get to electron microscopy, I wanted to make some notes on light microscopy. And there's a question here about something. I'm not sure if I quite understand the message about piano or whatever, but anyway, I'm going to make some notes here about light microscopy. And so what I've done is, like I said, I usually do this on the board. And so you can see I'm going to give you some notes about light microscopy. And then we'll talk about electron microscopy and fill in that part on electron microscopy. So first thing is light microscopy. And I mentioned that every microscope has some sort of illumination source. And so usually we're talking about some sort of visible light. For a light microscope, it could include sunlight, could be a candle, could be a light bulb, could be a laser. We have quite a few different types of light sources. And for lenses, we were using things like plastic and glass and quartz. And there are other materials, all sorts of composite and artificial materials that we're using nowadays. So before I get any further, I just want to point out that quite frequently I have a question on the midterm that asks, some sort of question asking about light microscopy and electron microscopy. So if you're going to talk about light microscopy, mention something about light, right? That's kind of an important aspect about what is going on in terms of how light microscopy works. So how are we viewing things on our light microscope? You can view it with your eye or a camera or something like that. And as we saw in these pictures, we can view things. What we're viewing at a light microscope is in color. So I'll put that. Maybe I'll just put. Sometimes it's not the right color because it's stained. But what techniques? So we have oil immersion. I mentioned fluorescence and confocal. Of course, I can't seem to spell today. I think it's U before O. There we go. And what was the other one? Staining is another technique that we can use in light microscopy. And what about the cost and ease? We will call this affordable, meaning that this is the kind of thing that we can afford at the college. A hospital could easily afford microscopes, even fluorescent microscopes. And we'll mention what affordable means when we're looking at electron microscopes. What's kind of the magnification? So we're looking at about 100 to maybe 2,000 times magnification for a light microscope. Usually about 1,000 times for typical light microscope. You add in fluorescence, you can kind of extend that to 2,000 or 3,000 kind of in there. And the resolution, just a second here, I'm looking for my notes. The resolution is up to about maybe 0.1 micrometers. That's a micrometer. So we can see things that are a little bit smaller than E. coli is really what we're talking about here. Okay, so if you don't get all that, that's okay. I'm going to come back to this in a minute. I want to talk about electron microscopes, and we'll compare what's going on with electron microscope. Like I said, what we want to do is we want to see things better. We want to see organelles and ribosomes and things like that. We want to see them a lot better. So let's talk about electron microscopes. So one thing that you may have noticed is that little formula we had in the lab manual last lab in lab 1 that it talked about punching in the wavelength and all that. So one thing, if you use that kind of formula and you punch in the wavelength, you can see that you can get better resolution at blue and violet light versus red light. But what if we were to use something outside of the visible light spectrum? And this is where the theory came up, basically in the 1930s, so almost 100 years ago now. People were like, well, what if we use some sort of other wave? What if we use electron waves? And so electron waves, it turns out, have a smaller wavelength, which means we could, in theory, see smaller things. So people worked on these for a while. There were some people at the University of Toronto, so a Canadian connection, and they wanted to make an electron microscope, and so they did. And in some ways, kind of similar you've got, here's on the left, you've got your light microscope on the left. And on the right, you have, sorry, I'm just looking for my pen. I don't know what I did with the pen. On the right, you have the electron microscope. So over here, you can see my light source is light. Over here on the right, for the electron microscope, I have an electron gun. So basically something that's shooting out a beam of electrons. My light microscope objective lenses, this could be glass or quartz or something like that. Over here, my lenses, it turns out, are magnetic fields. And so it's a little bit different, but kind of in some ways the same idea. You can't stick your eyeball into an electron microscope. You're going to zap it with some high energy electron beams, but you can use a detector of some sort, or a computer, or some sort of photography film in order to capture your image. You can see on the left, I've got a little mosquito. On the right, there's a little mosquito there as well. So let's talk about some of the details of electron microscopy. There's two types we're going to talk about. One is called scanning electron microscopy, and scanning, oops, I didn't mean to change the slide. So this is also called SEM, so scanning electron microscopy. And here's a photograph of the one that I used at the University of Guelph. And actually what you're looking at is the microscope itself is right here. So this microscope was purchased in the 1980s. It's about $100,000 for a new one. So you're not going to buy these things all the time. And what you're looking at over here is kind of like an old, it's used basically for focusing, but it used to be a bit of a computer. Somewhere over here that we actually had just sort of a desktop computer, you know, sitting there and running most of the functions on it. So I'll tell you a little bit about how this thing works. I think I have some images. There's some images I took of some bacterial cells, and you can see I have a variety of things here. And the images, they come out in grayscale. The one on the bottom here, this one here, I actually colored just using some software, and just to make them look a little nicer. So one thing you might notice with these pictures is you're looking at kind of a surface picture. So in this case, what you're doing is you're coating your specimen with some heavy metal. So usually gold is used, and the electrons are kind of bouncing off, and you're getting this 3D kind of surface picture. And here's some images, by the way. I kind of wish these were things I'd looked at. A lot of people will put insects and parasites and things like that under these scanning electron microscopes. You get some incredible pictures. I think the hookworm is one of my favorites. It just looks absolutely terrifying. So like I said, we can colorize these things. Here's some images of the virus that causes COVID-19. And you can see on the left, there's the original image, and on the right, they've colorized it, and it just gives so much more detail, and it pops out, and it's just much more visually attractive. So this is why we color these things, just because people like color, and color is a nice thing. Here's another colorized one. This is another virus. This is HIV. It's budding off of a white blood cell, and you just don't see it quite as nicely if everything was just in grayscale. Some people are really good at adding color. I'm not sure exactly how they did this, but just really pops out and looks beautiful. There's some E. coli for you. And some people get a little bit artsy with this. Here's somebody did this photo called Salt and Pepper. So that's a salt crystal and a peppercorn grain, and you know, pretty cool picture. So let's talk about how we do scanning electron microscopy. I'll show you some of the steps here. So if you take a look, there's a little mosquito. It's on the end of some forceps. And what you do is you take your sample and you put on a little platform called a planchette, and you stick it in this machine. I don't really know how this machine works. It's called a Sputter Coater. And basically what it does is it spits out gold onto your sample. How exactly it does that, I'm not exactly sure. It's not using very much gold, just a very, very thin layer. So it's not like it's super expensive. But you know, you stick it in there, it takes about a couple of minutes, and then you pull your sample out, and there it is. And you might be able to tell that it's looking kind of shiny. Then you stick it in your microscope. You can see it has this big durable door. And the thing is, what you're going to do is you're going to stick your sample in there, and it's going to be under vacuum. And the reason you put your sample under vacuum is that air has electrons. So we're trying to get out all the noise and all the interference out of there. And then basically you turn your microscope on, kind of like a traditional microscope. You're going to focus it using some knobs. This time there's more than two knobs. It's several knobs, but eventually you can get it into focus. And there's our insect head. And you can zoom in. I'll show you some pictures. Here we go, zooming in on some hair. And you can zoom in even more and you can see some exquisite detail on the hair there. And in fact, if you take a look, we're actually looking at individual proteins here, which is really, really very impressive resolution on that particular microscope. So that's scanning electron microscopy. And there's some more images. I just love these images. They're just really incredible. There's Giardia again, some human blood colorized to make it look more accurate to its actual color. And there's some human chromosomes, so pretty cool. And one thing that we can see that we can't see with a light microscope is viruses. So really zooming in to see tiny, tiny things. On the left there is a picture of a cell, and we have Ebola viruses are budding off of it. On the right, we have a bacteriophage. That's something that infects E. coli and kind of has a cool structure to it. So that is scanning electron microscopy. I want to talk about one other type of electron microscopy, and then I'll go back and we'll take some notes in that table I was making. So the second type of electron microscopy I want to talk about is called transmission electron microscopy, or TEM. So in this case, our specimens are still stained with some sort of heavy metal. The reason why we use heavy metals, by the way, is because heavy metals have a lot of electrons. So it helps us to see them a lot better. You don't have to. It's just much more difficult if you don't have a stained specimen, again, for the reason that we stain a lot of things under microscopes because it just helps us to see them better. So notice this first note, it says the electrons in this case are passing through. So they're transmitting through, right? So that's what's called transmission electron microscopy. And so the specimens have to be quite thin. And this will actually destroy the specimens if they're in the microscope for too long. And but it does work, and you get a different type of image. So here's what they look like. You can see the electron guns are often a little bit bigger, and these here are even more expensive microscopes. So we're talking about $250,000 plus, unless you get one used. I suppose maybe you could find them on eBay. I don't know. So I did some transmission electron microscopy. It took about 2 weeks of my life to get this image. It's not very good, so I kind of moved on. But you can see it's a different kind of image. You're seeing inside the cell with transmission electron microscopy. I'll show you some better images. So here's one from the textbook. You can see they've colorized it. You can see some detail in there inside of E. coli. You can see its DNA. You can see its membranes, and you can really, really zoom in. There, for example, is a couple of membranes. This is actually 2 membranes. So this is a phospholipid bilayer here, a phospholipid bilayer there, and you can actually, you can't see the phospholipids, but you can see that there is a bilayer and get a lot of amazing detail. Here's some more colorized. You can see some organelles. We can see the red things are mitochondria. The endoplasmic reticulum is the blue stripy things, and all those spots, dark spots, are the ribosomes. And of course, we can see viruses. So on the left, we've got pandemic from 10 years ago, the H1N1 pandemic virus. And on the right, we have the coronavirus pandemic virus. So you can see a lot of detail on them and the spike proteins and all those kind of things. So there's just a comparison. Transmission electron micrograph, scanning electron micrograph. You can see both looking at the endoplasmic reticulum, but different kinds of detail. So let's go back to our notes here and that table I was making. And what I wanted to do is kind of compare the electron microscope with the light microscope. So this time we're not using light anymore, or at least visible light. We're using electron beams. So we'll put that one in, electron beams, or electron gun, whatever you want to call it. all those things are basically accurate. What were the lenses made out of? The lenses were electromagnetic fields. Oh, magnetic fields. There we go. How are we viewing this? We're not viewing it with our eyeball. You can't do that. Like I said, we have high energy electron beams. So you're using detectors, and they will put the image up on a screen, or you can use photograph film. That's how we did it in the old days. Depending on the microscope you have, you might use one or the other. All the newer ones, of course, are using detectors and giving you instantly digital images, which is of course really nice. Color, nope, this is grayscale. Grayscale. Color can be added later, we'll say. So, and you don't have to use the real color. You can color any color you want, right? So what were the two techniques we were talking about? SEM, which is scanning electron microscopy. And we have TEM, which is transmission electron microscopy. Cost, we'll just say these are pricey. And it's not just the price of the instrument you're looking at. You're also looking at the cost of technicians. If you were to buy an electron microscope for your basement, would you know how to use it? No, you'd probably have to hire a technician to use it for you, and you're going to pay them the big bucks to operate it, or you're going to have to get trained. Scanning electron microscope, you could probably get trained in a couple of weeks. The transmission electron microscope, you know, it's a month or more of training, and even then you're not going to be very good at it. So what kind of magnifications are we looking at? So kind of 10,000 times to 1 million times. So that's huge. You're not going to get the 1 million times with a scanning electron microscope. That's more transmission, but maybe 500,000 times kind of magnification. And I can see over here on the left, I have spelt micrometer incorrectly. So let me add that R in there. And then what kind of resolution do we get? So up to about 100 nanometers. So that's 100 nanometers. Oops, not 100 nanometers. I believe it's 10 to 20 nanometers. We'll just say 10 nanometers. That's 0.01 micrometers. So it doesn't seem like it's a lot better, but it is quite a bit better. You're looking at a factor of 10. That 0.1 micrometers that I'm showing you for a light microscope is really just using some very advanced fluorescence. In most cases, you're not getting anything like that. And so this is for SEM and up to about 2 nanometers for TEM. Okay, so I'm hoping everyone got all that. If not, you can always ask me about it later, or you can review the, I'm taking recordings of all the videos of all these lectures, and you can always review the recording. Or send me an email later, I can always send it to you. I don't usually post these, it's just I end up with a lot of documents. I just want to post the basics, but if you want, I can email it to you, or you can review the lecture video recording. All right, so back over here, and just looking at the time, I realize we're almost done. So a couple of more things for you. I'm almost done here. Just want to show you some E. coli. So there's first one is a gram stain using light microscope. That's about the amount of detail you can get in E. coli. Maybe a little bit more if you use special stains. There's a scanning electron microscope image, much better detail, and there's a transmission electron microscope image. So you can see much better detail. So just to finish off, I just wanted to show you this. I found this on the internet a number of years ago. This is apparently the biggest transmission electron microscope in the world. And what you're looking at here is actually handrails. So this thing here, that somebody would be walking here. So here's a little guy right there. And I don't know how much it is. You could call Hitachi for a quote, but there's a technician. He's probably making a lot of money because he's probably one of a few people that know how to operate this beast. But and I thought I'd show that to you. If you can't afford one of those, I found this on the internet a few years ago for $50,000. Don't know how old it is and what condition it is, but you can buy used ones out there. So anyway, let me just see where we're at. This will be the last slide. So remember, when we're talking about these things in terms of the functioning of what is going on, I made that table for you to compare how electron microscopes and light microscopes work. And remember, if I'm asking on an exam, don't forget to mention what the light and the electron parts, what does that have to do with the whole thing? Don't forget that. I'll show you a sample midterm question next day, but we're out of time. So thank you all for coming. I will see you on Wednesday via Zoom. And if you want to get a hold of me beforehand, you can always set up a meeting with me or try giving me a call or an email. So that's it for today, and have a great day.